And actually, I think we are at 2 o'clock, so I would like to transition to the full panel discussion. So if Rob and Sean, you guys can bring your videos back on and join us, that would be great. And I think there's a few questions here that are aimed, maybe aimed at all of you. Let's see who's willing to answer them. Um, One I'm gonna pull down, not the very top one, but the third one down, which is how do you combat the risk of duplication across different repositories? Because I think I have an answer for it, and I'd love to hear your answers for thinking about that problem.
Well, I can take a stab at it. This is Rob. That's been the whole premise of having Proteome Exchange. We don't really want to have duplication across resources, although we know we do have that. The whole idea is to alert the other repositories that that piece of data is there. Now, that data can reside itself still in that same repository, but a subsequent analysis of that data and its results can then be shown on a different website. So we do that ourselves at Peptide Atlas. We take data from, say, Pride. We take some data from Massive. It's there. Majority of it tends to be all in Pride, and we reprocess that ourselves and we present that data. So that's a different aspect of the data. But the raw files can really maintain itself there. And so when we produce data results, we also point back to where that data was in an initial repository. I suppose in that second question there, where it says, "Are you expecting researchers to cite the resource, either the repository or the user?" I think the best thing is to always do both. If you know that the data set was done by a particular user and they have that published, cite them, but also cite the repository where you got that from as well, because then that will allow people to access that data again of what you've seen.
Other thoughts on that? Sean?
I guess I can have a go too, because we do understand, especially when it comes to our data repository, that duplication is a potential outcome. I will say that we do alert users to the fact that it's possible. There's not a whole lot we can do about it, but we do alert users within the data use agreement that duplication is possible. I will say also too, though, that there are so many research questions. It's a low probability, but it has happened in the past that we're aware of. And I guess also too, regarding the question up above, regarding expecting researchers to cite a resource, that's also included within our data use agreement, that there's a little acknowledgement that we have users put into their publication. Do you want me to do the top one too, as long as I'm here?
Right. That reminds me a lot of the way the clinical community handles it. Oh, sure. That would be great.
So I'll try to do that one quickly. There's a bit of an algorithm there, just because there are several thousand names within kind of our tracking system there. You don't want to just search on everybody's name. So there's a bit of an algorithm there, but so it's a lot of PubMed searching. We do it 3 times a year. It does take a long time, but we do a lot of PubMed searching, review the hits, and then if necessary, we read the paper and try to figure out just what exactly was the source. So it's time consuming, but there's a bit of an approach to it as well.
Right, and I think we heard a bit of that this morning as well, making sure that good citations are present and trying to encourage people to cite as well. So actually, I'm surprised that neither one of you mentioned consistent identifiers, and I guess I like digital object identifiers as ways to track things, particularly between repositories. But you didn't mention that, so I'd love to hear why you didn't and how you think about just the IDs of either specific data sets or specific proteins, specific clinical trials, whatever it happens to be, and how you do go about making sure that citation happens.
Yeah, so for us, the repositories do have a unique identifier. So if you're going into PRIDE, you have a PXD number so that that identifies individually that data set. There's also the other repositories, Massive, will have their own unique identifier within that as well. So those identifiers there. I did mention PXDs, but not explain them. So sorry that I missed that out. The other effort that's actually going on at the moment is to try and provide individual spectrum unique identifiers as well. And so here you can at least provide the spectrum aware identification of that particular peptide, which then infers the protein that you've got, has a unique identifier so that that can actually be tracked and you know exactly which run, where it was, and what that identification spectrum was there cited in the first place. So that's an ongoing effort now that's going on to provide these UISs, and so that is very much on top of the mind of the repositories and making sure that we can link these information across, and that's a very valid point.
Great, thank you. See here, I think those top two questions have been answered at this point. Let me just go to the whole list here. So there was a question, and I think it's to you, Sean. How are you distinguishing between publications about data generation versus secondary use? I'm not sure I completely understand that question, but maybe you do.
Well, in terms of data generation, I guess I'm not entirely. Oh, okay, I see the question there. Are you distinguishing between publications about? Okay, so there really aren't publications that I'm really aware of about data generation. So in terms of our data repository, it's all going to be related to secondary use. Now that can be in different ways, whether it's kind of an empirical example of a statistical approach, or kind of a research article or letter. But I'm hoping that answers the question.
Well, and I think maybe the other way to frame that, and I think both of you say this, is you want people to cite both the data generators and the data set, therefore the data generators, as well as the repository. So maybe that gets also at this question.
Okay.
And I think that's connected to the question up above, and that's how do you know the reason for a particular data access request? And I guess that's perhaps going at Brian's question of are requests made through a data access committee? Do you use dbGaP, that kind of?
So we have a fairly simple data request form.
And that's how you.
So we have a fairly simple data request form that the user completes, and there they do describe the reason for their data access request. It's not a merit review or any kind of a committee review. It's really kind of appropriateness of the research, is making sure that the purpose is consistent with our data use agreement in terms of the controlled access data sets that we have. So when it comes to like that reason, and especially like that one graph that I had, so we end up coding it based on what's described within that data request form.
Great, thank you. And I think back to you, Rob. Oops, we just had them hop around here. And I think you already addressed this, but maybe this is an opportunity to say it perhaps a little differently. Do you see any particular hurdles in linking data across several repositories? And again, I think you addressed that a little bit, but maybe not exactly in that way. And I think in particular, they're asking for recommendations for others that are undertaking that kind of, I'd say almost a federated approach to making consistent data available to people?
Well, yeah, there's the Proteome Exchange Committee. So there is. I would attend the Proteome Standards Initiative meetings. These meetings where all of the repositories come together anyway. There's many different efforts, and then also I would, for the protein type repositories, I would also go to the Human Proteome Organization meetings, which are held every year at the third quarter of the year. So this year it'll be in Stockholm in October. And that's where these sorts of things are discussed. Most of the repository people are all there. They're all exchanging the ideas. They're all making the connections. And so that would be the best way. But in the meantime, I would just get in touch with the major people that are doing that. That would be Eric Deutsch here from ISB, who's heading the Peptide Atlas. Henning Jacob from EBI, he would be the best person to talk to there. And there's many other people at different repositories as well. We have Massive. There's also the Panorama databases here at UW. All of those people are accessible online. They're all listed on their particular websites, and I would just go there. So,
great, thank you, Brian. I want to have you address this question if you're willing to, and that's, how do journals like Scientific Data, so the Nature journal, fit into the ecosystem of data sharing, and is that a useful approach? And if it's not, how would you, what would your suggested alternative be?
Great question. So I know a little bit about Scientific Data, the journal, and I would say that there's a number of different benefits that having such a journal brings to the table. One of them is to have a journal that's focused on presenting the various issues. So, for example, I happen to be familiar with an article that was published in Scientific Data that talks about the struggles of trying to access publicly available transcriptomic data in the pediatric cancer world. I don't know how many journals would necessarily publish such a thing, but I think it's a very important topic, and it proposes solutions for how things could be easier for reusers of the data. And if that's not important, I don't know what is. That's such an incredibly important area of research, and I think that the journal brings a certain vantage point that is very valuable for the research community, and I think having data publications makes a lot of sense. They're a citable object with the DOI. I like Scientific Data as part of the ecosystem. I think it's very useful.
Right, and I think going back to some of the conversations this morning, and just to remind everybody in the audience that all the presentations will be made available after today. And in particular, if you check the Twitter feed looking for the hashtag NIH data metrics, it should all get posted there, so you'll be able to find all the presentations. But there was a discussion about citing data as a really important part provided. So it sounds like you're concurring that that publication or that journal is one way to do that.
I think so.
Yeah. So there's a question. I think this is a philosophical question because I don't believe there's an answer to it, or at least not a clean answer. And it's actually, I'd say there's one part of it that is perhaps answerable, but then there's another side to the question that I don't think is answerable, and that's, how do you measure the size of a community that a repository serves? And then I think the part that's maybe harder, although it's well written here, is that what's the expectation for how well a particular repository needs to serve a given community? So there's a for instance, of if you get 200 new users per month, is that low or high? And again, they're putting it in context of the size of the scientific community that would be a potential user. So again, I'd love to hear your thoughts on either as a user of those kinds of repositories, as a owner of those repositories or as a funder of those repositories, how do you think about that?
Everybody's looking at me, I feel like. Which is a great question, and it's one I actually hadn't even thought about, to be honest. You'd have to think that with an organization like the National Heart, Lung, and Blood Institute, that's a fairly big community. I don't even know how many grant applications we receive in a particular year. So, I would think but of course they make their money from new grants, from creating new data, kind of a thing. I think it's a great question, but I don't know what would be high or low or how to set expectations. I kind of feel like, though, this is just kind of a feel like, that we would have to be regarded as low, I would think. We add somewhere around 120 investigators a year, but I mean, it's a huge community. And, to be honest, I'm a PO for one grant, and I was talking to my grantee, and he was going, "Yeah, I found this BioLink, and there's all this great data in there. Why didn't I know about it to begin with?" I was, "Oh, I don't know. You really should do better marketing, I guess." So I think it's how well are we kind of reaching our community, is probably a good question. But it's something I haven't thought about, but I'm now starting to think about.
Right. So it's the idea. There's a bunch of marketing pieces there. What's the penetration of knowledge in your potential consumer group?
Exactly. Yeah.
That's an interesting question, and not usually the way that we think as scientists, is it?
No. If you build it, they will come. Well,
Right.
I guess you got to tell them too.
Yeah. So Rob, I think you run a repository that's really goes across lots of different disciplines. People just have to look at protein, though. So they're all asking all kinds of scientific questions. And how do you make sure people are aware of what you're doing?
Yeah, look, I think Sean really touched on it as well. Marketing is one of the key points to getting it out in front of people's faces. But at the same time, scientists are not trained as market people. So it is a hard thing. You need to step out of the academic world into the commercial world and start to learn how to do marketing. But I think one of the things that does come to the point is that if you do have a resource, it's beholden on you to actually get out there and talk about that. We do that as much as we can. Now, our marketing, I would say, would be woeful as compared to commercial companies, and we'd always love to work out how to do that. But there's no funding to do that either. I think if the NIH was to look at how to fund sustainable resources, then a component of that would be to fund marketing too, and really make that accessible. I think the bigger repositories where everyone looks at, such as EBI, they have a fairly good track record because they hold courses there on many different aspects, really on a weekly basis, and that's something that is part of the funding system in Europe. The US doesn't really have that, so I think having some way that the NIH could, goes back to this question below, is how to tweak the peer review, is help the repositories do that kind of stuff so that you can get out to the people, and then you can start to measure how many people are really picking up the resources over time. I think at the moment, a lot of it is word of mouth. They see it at a conference, and then someone else tells them. But it's bad because it's, as Sean says, if you build it, they will come, but not necessarily if people aren't shown. And most of the people need to be spoon fed too. Everyone has their own research focus. They want to do their own things, and having to actually look outside and look at data resources is another investment in time, and that's something that usually people find that difficult to do. But when they see the value of what's out there, then they can actually make sense out of what they're doing. And I think it comes back to marketing. It comes back to how do we do these things in a sustainable manner.
So, Brian, I'm going to put you on the spot because I think actually a lot of people have heard about the Symbiont Awards. And obviously, you don't have a marketing budget for that, but yet somehow that's gotten out and known. So, how have you done that? And how do you think about that as a I don't know, it's not necessarily a problem, as maybe it's an opportunity.
Great question. I would say that the Research Parasite or Data Parasite Awards are perhaps better known, and I think that the name was a stroke of genius, and so I think it's gotten a lot of media attention. The Symbiont Awards, I think, have gotten some attention as well, and I think it's been basically organic. It's been talking to people about it, tweeting about it. I do have a Twitter account and do share information about the Symbiont Awards on the Twitter account. I think some of the winners have been instrumental in advertising them, and I think I take opportunities when I can to make sure that others are aware that we're trying to shift the culture, kind of flip the script from people feeling that it would be advantageous to hold on to their data and instead maybe try to create an environment in which people feel that there are advantages and recognitions that are due them if they share their data, and not just share it. And I see one of the questions here has to do with accounting for the quality of metadata. It's a key point. Not just share it, but share it well and share it in a way that is reusable. And that's one of the reasons to address that question that's on the screen there about how to make use of, or how to rather judge the quality of the metadata. I think there's only one way to do it, which is to look at the metadata and then in conjunction with that, look at whether other people have been able to publish papers of high quality using those data. If nobody is publishing, maybe there's difficulties with the metadata. And of course, there are other ways that people check metadata, and sometimes people have found that metadata have been inaccurate. In fact, that data, the Nature paper that I was talking about earlier, the data, what's the name of that journal? Anyway, the data-focused journal that Nature has, there is a paper in there about the difficulties of getting pediatric transcriptomic data related to cancers. And one of the things that the investigators report is that some of the metadata describing RNA turned out to be DNA or male-female confusion or other things. So it's absolutely the case that some of the metadata labels are wrong, and I think there's not a very simple way to address that problem. So I think it's a very good question, by the way. But those are some of my thoughts.
That's great. I'm going to ask you a follow-up on that. And actually, I'm not sure who will answer the follow-up, but something I've seen is that what you're getting at is we really need to have a data ecosystem so that as people download a particular data set, as they reanalyze it, as they actually improve the metadata, there's a place for them to put that so that it becomes. And that's turned out, at least in my experience in the time I've been doing this, to be a remarkably difficult thing to do. And I'd love to hear one if the, I guess this is really for Rob and Sean, the repositories you have, do you feel like it's easy for, if you will, the folks that are downloading those data sets to be able to express their findings? If you will, give a review, and it's actually a very special kind of review because they can also contribute back data about the analyses that they've done. So are we ready for that? Is that, are we there? Has it already been happening?
Everybody looking at me again?
Sure.
I think you guys both, I'm gonna make you both answer that, but.
So we have thought about, of course, when somebody, downloads data, one thing is about, with the BioLink and the web portal that we have for the data access, is there's of course, opportunity to post any questions and all and give feedback and all those kinds of things. So that part kind of gets easy to handle. We do try to address user questions as much as we can. The other side, which I think, let me know if I'm kind of wrong in how I interpreted this, but of course, when users do download data, they end up transforming it a bit to fit their purpose. And so that might be, essentially creation of some sort of derived or new calculated variable that perhaps could be meaningful to others as well. And we've thought about asking for that, but thought, kind of went against that approach. No one has really offered to essentially kind of provide either code or kind of like this new calculated variable. And we don't require it with the biorepository as well. If you do some new analyte or something to redeposit that. We haven't required those sorts of things, but they do come up in conversations. I'm not sure if I can really express why we haven't tried to stress or ask for it, but we haven't.
Rob?
Yeah, have we been thinking about it? Yes. Have we got something in place? No, as a short answer. The attempt to try and put value back to the researcher really has been the Omics DI component at the moment. You can read about that. Currently, it's in bioRxiv. There's a paper you can pull down there. At least that's one attempt of trying to put value back to the researcher who's taken data and reanalyzed it and said, okay, what can I get out of it? Are we taking those reanalyzed data sets and putting them up at the moment? No, we're not. That's something that really needs to be worked on. Part of this has been trying to develop this universal spectrum identifier where you can actually go back in the mass spectrometry data and say, okay, here is this spectrum that people are debating about, and at least you can pin it to that exact same piece of data, and no matter what the interpretation is, getting that interpretation up really, it's at the moment it's just in a publication, and you'd have to go back to that publication and filter it through. So are we actually taking those new reanalysis through yet? No. And I think that's something that really needs to be worked on, and it's a goal that would put a lot of value onto the resources as well, because new software is being developed all the time, new algorithms, new ways to interpret data, because none of the data is very clean either. The newer approaches now are producing multiplexed spectrums from many different peptides incorporated in them, so the algorithm can get fooled very easily. So we're in an infancy of new styles of way of doing the work and getting the raw data, and so we need to catch up again with the algorithms. But ultimately, I think that goal of putting value back to that reanalysis of data does need to be done, and I think that's a good goal to have. Part of it is a work in progress, but not the whole part. And I think we need to look at ways in which we can. One way is to at least fund something like that to be done, and especially just in the proteomics community, but it would also be relevant to the other communities too.
Right. I think that is actually a goal for the various NIH-funded data commons initiatives. Now, I was involved in the NCI Genomic Data Commons, and one of the goals that we clearly had was not just providing data sets with well annotated analytical pipelines, but then as the community used them, be able to annotate back what was either wrong about them, what they found that needed to be improved, and then just getting those usage metrics back. This was a particularly valuable data set or genome for this kind of question. But that turned out to be remarkably difficult to do, and I think you're both indicating that that's been your experience too. So there is a question here for you, Rob, and that was based on your comments about funding. Do you have any policy or outreach recommendations that you'd like to use to reconcile the reality of resource maintenance and community expectations for resources?
Well,
I know that's a tough one.
Yeah, so for outreach recommendations, I think you know, there's always a cost in FTEs on employing people to do the work to maintain that, and then there's also the cost of just housing it as well. So none of these things come for free, and someone has to pay for that in the end. Maintaining just a database in itself is probably a low cost thing, but it still takes people and equipment to do. But if you're wanting to develop new aspects to that, that's when it starts to become expensive, because you're in an environment where you're competing against many of the bigger companies these days, the Amazon, Googles, Microsoft, all those kind of things. We're throwing a lot of money at talented software engineers that take it away from science. But you're still able to do things, but you've got to compete in that environment. So some of the things that needs to be done as a policy is to look at, okay, what are these big data sets or these big data repositories doing? How well have they served the community, and are they in a community setting? So have these repositories already been part or formed a part of a community in which the databases are federated? They have access guidelines in which people can do things fairly easily, and then you can start to say there is some resources that need to be maintained, and the community expectations for the very basic one is it available? And we saw that example when there was the NIH funded effort that was out of University of Michigan that was also a repository. And when that funding went away, it died overnight. Yet the journals were also supporting that repository, saying that we need that data for people's submissions. And it was the very first infancy of that. And when that resource suddenly disappeared because of no funding, the people were let go, the computers were turned off, the journals didn't really know what to do. And so they had a moratorium for several years of, well, we just can't expect people to have them in repositories anymore. So the community expectations are that, for one, it's there, but then the usability. How well can they access the data? How fast is the response? And what is the richness of that data in there? And I think as the Have I been on mute this whole time?
No, I just was suddenly muted as well. I don't know what happened, but we heard you right up until then.
Right. Yeah. So the expectations are, if people see these data sets as important, and just to toot one's own horn, the Peptide Atlas has been supporting the human proteome effort on a voluntary basis for all these years. That also counts to why these resources are available. And on top of that, you've got PRIDE and Massive supporting the storage of this data. So this is petabytes of data that is stored, and someone's got to pay for that in the end as well. And then you've got an Uber resource as well, which is NextProp, which takes all of that aggregated information that's highly quality controlled by really the whole community, and says that this is something that we need to ensure that the identifications that we have is correct, and then we can use that information for all the biological studies that we're doing. So where does that protein being seen? What's it identification like? Where has it come from? That then lies, this community expectation. So I think, the recommendations here is that we have a system in place where things are being federated through these various initiatives, whether the Proteome Stands Initiative or Proteome Exchange, that we have capabilities, and the community expects them to be there. So how do we resource that is the bigger question, I think. And we all understand the difficulties in funding, but there has to be a concerted effort to look at that in the long term. And even the NIH had an effort on that themselves, which they shut down after about 2 years. And so, this is another loss that had to be reconfigured and absorbed elsewhere. But ultimately, if we expect these things, then we need to have the right resources.
Well, and I think that one of the goals for this particular workshop is to come up with ways of measuring the value of data sets, the repositories, to the community and to the way we do science. So I think that is part of what we're hopefully doing today. It won't be the end; it's the beginning. I want, though, turn a little bit sideways here, and again, Brian, I'm going to go back to you and think about a world in which we can make a lot more of our data sets publicly available, because we've actually resolved some important social conversations around privacy, particularly looking at clinical data sets. It's interesting. Some people figure out how to make them publicly available, but most of us don't. I'd love to hear your thoughts about how we work more. And this is a tangent. How do we get the right kinds of consents in place? And maybe it is also the right kind of mindset so that we can make data more freely available.
Great question. There are people who are spending a lot of time thinking about how to do the consents that will enable data sharing. And people who come to mind are John Wilbanks and the other colleagues, Brian Bott and others. And there are meetings like the Pacific Symposium on Biocomputing where these type of topics are coming up. And so I think that there are people who understand these issues deeply. And one of the things I've talked to John about is, can you make any of this knowledge into a kit or into something that's dispensable to others so that people can understand those things? And I think he's interested in doing that. There's limited bandwidth, but I think to draw on the expertise of people who think about that question all the time is a very smart thing to do.
Right. Well, and actually, I think just talking about John Wilbanks very briefly, he does have, and Sage Bionetworks has been developing the e-consent and the dynamic consent modules. Of course, that's deployed to all of us now, and I think they're really finding out how do we do that at scale.
Right.
But I'll say there's still a line between gaining access to those resources for most of the community and making them truly public.
Right.
Thinking through that part, again, I find it fascinating that the 1,000 Genomes Project, anybody can download any of that information at any time. There's no barrier whatsoever. But in the cancer world, we haven't figured out how to do that, even though we believe that if we're looking at germline data, somatic data, it really isn't identifying, but we still don't make it freely accessible.
Right. I think it's a long discussion. I have a paper that'll be coming out I think soon with Casey Green and colleagues where we'll address some of these topics in more detail. But I think you're asking really profound questions. There are these issues about what might not be identifiable today becoming identifiable in the future. But I understand your point about if you have all these driver mutations and perhaps many sort of follower mutations, that the somatic tissue could be very different from the germline tissue, and you may not be able to draw any inferences about who that person is. And so I think my guess is that more and more the expectation will be people will find ways to share. But as I mentioned during my comments, I suspect the most efficient way to make that happen would be to set up the incentives such that people are aware that their data sharing history will be judged based on the case that they make, maybe not in a single metric, but perhaps the case that they argue in a section of the grant application. that would then be judged by their peers. I think if that happens, you could really change the incentives toward people wanting to figure out how to share well, share in a way that other people can reuse the data, which means having good, high quality metadata or a data portal that is easily used. And we might see more instances of people sharing their data. In fact, I think it'd be inevitable that we'd see more instances of people sharing their data and sharing it well if doing so was a criterion on which their future funding chances hinged.
Great, thank you. And there's actually there's two questions that are pretty similar, and I'm going to ask you somebody to address them. And one of them is: have you measured the impact of requiring data access requests on the usage of data? And I have an anecdotal evidence for that, but I'd love to hear if either Rob or you, Sean, have tried to quantify what some of those barriers to requests actually do for usage.
Rob, you have to go first this time. Okay, I'll go. So have you measured the impact of requiring a data access request from users? That's hard to measure. I think one thing we do is, we're the stewards of this data, and so it's all human subjects. So we take that responsibility fairly seriously. We do have some data sets. They're mainly geared toward kind of didactic purposes. We call them teaching data sets. There's also like a true public use where you don't have to do anything other than, kind of push a button and download. And, those do have a lot of action to them. Clearly, there would be much more, I guess, usage or activity if there wasn't kind of a controlled access approach. But for us, I think our stewardship responsibility makes it imperative, and it's because it's human subjects, makes it imperative that we do have kind of a controlled access approach. So I'll stop there.
So it's hard for you to know what it would be like without that.
Right. I mean, you'd have to figure it would really go up quite a bit. The bar is not terribly high, we feel, but you do. But it's hard to know whether you get, kind of useful utilization of it. Whereas with people have to cross at least a few hurdles, they're more inclined to have that kind of clear need and purpose.
Rob, would you have an answer?
Rob is on mute, I think.
Rob, you're muted. Rob, you're muted.
No, I'm just not there on the protein side. Was I muted again? Sorry.
Yes. You were. It's okay.
Oh, sorry. So what I'm saying is that the protein world is very much different than the genome world, and there is no bar there at all for the data access, and so it is very much an open slather for people to have access. So we don't have those difficulties at the moment. Having said that, though, as we're moving forward, looking at particular human variant data in the protein field, then that starts to also raise all of those issues where you now need to look at specifically, can you identify a person based on the protein data that you've got there? Those data sets really aren't there that close yet. Most of the samples that are actually done, though, are already de-identified as well anyway. So that also puts another barrier to putting a data access request there as well. So at this stage, for proteins, there's not that requirement. Not to say that it's not there, but ultimately, I think that since the genomics people have already done that, you would just lend off of the same thing that's there. So why reinvent the wheel? You guys have already done that, so we should just copy that.
Yeah, so I can give a little a informal example, and that's it for the Cancer Imaging Atlas, so TCIA, and this was done by Paula Jacobs back when she was the director for that program. There used to be a requirement that before you could download the images, you had to register, and they removed that registration requirement, and the volume of requests and access tripled. And if folks are interested in that, I don't know if that's ever been published. And at the risk of inundating Paula with email, her email is public at NIH, and you could send her a note to find out more about that. But I frankly, I was shocked that something that small, where it wasn't even a data access request, it was really just registering, had such a large impact on reuse. That's different, and I think we want to get to this question. Use is different than impact, and how do you go about measuring impact on the usage of the data? So that's just kind of rephrasing that question about the impact of requiring data access requests to actually measuring the impact of a given use of the data. Again, I think we've gone over that from a citation standpoint, but I'd love to hear other thoughts about how you actually measure impact and seeing usage.
I think from a usage point of view, I think the best example has been when certain data sets have been put up and interpretations made that the data has been refuted because of the difficulties in analyzing very large data sets and being able to control false discoveries. I think what was published in 2014 were two papers on the drafts of the human proteome, and the reanalysis of that data showed that it was much less when you controlled in a quite considered way the false discovery rate, and so it was no different than what the Human Proteome Organization had already put out. That has a huge impact. Now, the other difficulty that you have is that when many people will take data and reanalyze it and find out that the data is maybe not what they expected it to be, or could even be better than what they expected it to be, the negative results will never be published. Many people feel they don't want to talk about that kind of stuff. They'll just say, okay, well, I won't use that data set. But if there's something that comes out of it, they certainly will publish that. So there's always this issue of what can be published and what people think about it. But I think ultimately the impact has been that the community then sits back and says, has this data been analyzed properly?
Right.
I think that point that Rob raised about what's effectively a file drawer problem is a great point. One I hadn't thought of before. Yeah.
So Brian, there's a question just came in that I think is wonderful, and I'll say that personally, I pushed for the creation of a data sharing index for a while. I think it's hard to do, but the question is really, let's help study sections use your idea. How would NIH tweak peer review so that your proposed reputation for sharing score would actually be used in helping them in funding decisions? Even though that's a no word for study sections.
Right. Well, I think
evaluation of the proposals. How's that?
Yeah. Right. The point that I was trying to make during my comments earlier is that I think that the Alex's Lemonade Stand Foundation has done a really good job of doing exactly that. And so I would hope that NIH might take a look at the way they've implemented this. In that setting, it's more of a rubric than a score. I think there are some limitations. I know people have proposed an S index, a sharing index, tied to DOIs and publications, which of course is a little bit limited because if what you're sharing is a cell line or a mouse or something else, then somehow you're not going to have that captured in your sharing index. But a sharing index rightly would capture other things that you're sharing. So what I like about what Alex's Lemonade Stand Foundation is doing is they're simply asking the applicant to make their case about how they're sharing with excellence, in essence, and then they're asking the committee members to assign a score 1 to 9, like you would assign it for other criteria, and it gets considered, in that gestalt that becomes the final score for that person's grant. I like it because I think it's not trying to reduce something complicated and multidimensional to a single number, and it asks the what one burden associated with it is that the people who are applying then have to do a little bit of legwork to figure out how people have been using their work, maybe more than a little bit of legwork if people are using it a lot. But I think if people are using it a lot, that legwork should be rewarded by the study section, who should give extra consideration to funding that individual by virtue of the fact that when that person gets funded, not only do they publish papers, but other people publish papers using that data set.
Right, and I'll make a plug for a number of the different NIH study sections where I do hear, when I've sat on those study sections, the discussion about how openly is either data or the models made available to the community. And again, I think people know at some level how good others are at sharing their, the fruits of their work. So I think making that a little more consistent and frankly an expectation is very valuable.
Yeah, I would make it a criterion.
Yeah. Let me just go see if there are other questions that have come in here. So this is a nice question, because again, I think we've discussed this. Rob, it's to you, but I think it really does hit every repository. And that's that do proteome exchange repositories commit to preservation of their data holdings? And I think there's an example of things that you gave of things that's going away. And is there a minimum preservation standard that you want all the members to adhere to?
Well, I don't think there could be any preservation standard put forward, because ultimately someone has to fund this, and even EBI is at risk of disappearing as well. If they have NIH funding, they also have EU funding, but if that disappeared, then they would disappear too. And this is an ongoing problem. I don't think that you can ever have a guarantee that the data is preserved. I think the hope is that with a bigger community support and a well organized resource, that these things stay up. And I think with us, we've been going on a shoestring. But that's because we primarily reside on NIH or NSF funding. So this is something that's always at risk, and no matter which repository. I think Massive with Nuno Bandera as well is also at risk, because he's on NIH funding, and he was started up because the University of Michigan one actually shut down, and the NIH suddenly realized a year down the track that was a mistake. And so that's when UCSD got a contract. But, everyone's always at risk of disappearing overnight.
Right. Well, we saw what happened now years ago when Swissprot lost their funding, and they went to pharma to get at least partially covered. And then that really limited access to a lot of academic researchers to Swissprot. Fortunately, that's all reversed itself. But it was again, I think it just highlights how incredibly important that funding piece is.
Yeah, and that's the idea of Proteome Exchange as well, is that at least the data sets are indexed from one side to the other, and there is some level of duplication, so that you do get aware of which data set, if it's duplicated, which one's closest to you. So it has that kind of mechanism that you don't have to go drag data from the other side of the world, that if it's in a repository closer to you, then you get that as well. But ultimately, are all the data sets preserved? No. So I think in an emergency situation, there is probably some capacity to take all of that data on, but that is still up to the repositories to do that if they have the funding.
So we're coming up on the hour here, and there's one last question that I'm going to throw out there that's on the screen here, because I think it's a useful closing question perhaps, and that is if we move to a user pay model. So I presume that would mean that if you want to access to a particular repository, wouldn't that really mean, and of course, if that comes down on people with grant funding to do those micropayments, wouldn't it still ultimately be the funding bodies who pay for these resources via the individual research grants? So it's a little bit of a provocative question.
I'll take a quick stab at it. I think that would be a problem, because you got to remember that the PI of any grant has sole rights to do whatever he wants with that money as long as he answers the question. So if he's put in there, I'm going to pay for a certain component, but then reallocates that money to something else, then there's no guarantee that payment will be made. I think that it would have to be a different model in which those vagaries of interests would be taken out of that, and so it would be guaranteed to come through. I think there's capabilities of looking at that within NIH or NSF funding, but at the moment, because the PI just answers to the program officer and says, "Oh, I've done this with my money," there's no guarantee that the budget allocation actually comes to the repository.
Ryan, what are your thoughts on that?
I'm sorry, I didn't hear whether you said Sean or Brian, but
Brian.
oh, well, yeah, I see that this is going to be challenging to move to a user pay model. I think as an end user of databases like Rob's, I think it would be quite a burdensome thing to have the user pay for access. And I think it would probably diminish use, and these are important resources. So my inclination, although I can't make it happen this way, but my inclination would be to make sure that valuable resources like Rob's stay funded from a federal grant directly.
And Sean, since you're at NIH, I'll let you answer that last.
Okay, thank you. Much appreciated. I would have to say, yeah, ultimately, by and large, it's going to be the funding body. But I've agreed with Brian and Rob there. It's not really a viable model, especially if your goal is to kind of maximize the investment the institute has already made in collecting that data. A user pay model, detracts from that quite a bit. So, just from my own perspective, the kind of centralized long-term commitment, something that the way NHLBI has approached it is the model to really pursue, and it has to be kind of an institute commitment. There are various kind of funding mechanisms that do these types of things, like the R24, the U24, but the institute has to support that. There's no guarantee of a long-term commitment. So I think that the approach we've taken is probably the one that in the end works.
Right. And well, I will say, to be fair, when I read that question, I really think it's a statement the other way around. If we move to a user pay model, in the end, the cost to the institutes are the same.
Or higher.
And potentially or potentially higher. And actually the impact on the momentum and the velocity of research would be pretty negative. And again, I think you've all said that. So with that, I think we are at 3:00. I really appreciate all three of you, both your presentations and then your participation in the panel discussion. So thank you all.